Name is still the structure and function of large biological molecules. Oh yeah, we do. Yeah, so we're finishing chapter 5. So far in chapter 5, we've gone through just a whole bunch of different tables, right? Right now, as you hear my voice in the future when you listen to this recording, what's on the screen is a table right out of the textbook, but I'm not going to draw you another table on the board. I just want to talk to you about something we've already talked about, but give you some specific examples. Okay? Specific examples. So something we've already talked about before is that these biomolecules basically come in monomers and polymers. Now, most of the lipids are monomers, and there's a whole bunch of different types of lipid monomers, but most of the lipids that are active in living organisms are monomers. There's only really one type of lipid polymer. That's not true of the others. Okay, that's not true of the others. In fact, you cannot have a protein monomer and have it be active and do anything other than be used for energy. Okay, you have to have a polymer. And with carbohydrates, you can have monomers, but most of the time it's stored as polymers. And then same thing with nucleic acids. There's really only one thing you can do with a nucleic acid monomer, which is called what again? A nucleotide, and that's to use it to store energy. If you're going to do anything else, you have to have a polymer. Okay? So I want to talk about some specific polymers, and then something we also talked about is that even though if you produce these monomers chemically, you tend to get half of the left-handed, half of the right-handed, right? We talked about that. What do we call those types of isomers, where they come in left-handed and right-handed varieties? Yeah, Katie. Yes, mirror images, but what's the technical term for them? Enantiomers. Okay, they come in left-handed and right-handed varieties, and in living organisms, you tend to only have one of those forms be biologically active. Carbohydrates are a little bit different. So I want to talk to you about some major carbohydrate polymers. And these are either structural or for energy storage. Okay? So carbohydrate polymers. Carbohydrate polymers. First one, starch. This is a glucose polymer, and it serves the purpose of being energy storage.
Like cornstarch?
Oh yeah. Yep, like cornstarch, energy storage in animals and seeds. Okay? So starch is a glucose polymer, and its function is to provide energy storage in animals and in plant seeds. Gosh, that was terrible. All right.
What were you trying to do?
I was trying to get it to stay on the table, but it just went plump. I know. If you don't attempt difficult things, you'll always reach your goals. Right? And you know what? The only thing that's worse than not getting everything you desire. Getting everything you desire.
Oh. That's deep.
Yeah. So, anyways, thanks. All right, so starch is a glucose polymer that functions in energy storage in animals and seeds, which is why we've got cornstarch. But then, bless you.
Why did you point to me when you said
Who said corn? Wasn't it you that? Oh, it was Isa.
No.
Well, I thought it was you because it came from
I
here.
was like, are you trying to say something?
No, oh my gosh.
You're a cornstarch.
So I felt really awful about calling you all terrorists on Wednesday. So I want you to know.
What we were talking about.
Well, because you guys were trying to disrupt the class by continuing to come up with other ideas.
Oh.
And so I called you all terrorists. And I'm sorry. This is one of these peacemaker opportunities. That was
We
wrong.
forgive you.
I'm sorry.
Emma is
Okay.
most.
All right.
Yeah.
That's so messed up.
No,
I'm
Middle Eastern.
I understand that.
You're just staring at me. Okay, I'm innocent.
Yeah.
That's
Okay. So starch, energy storage in animals and seeds. Sorry, I was thinking through. Oh my gosh.
No, are you gonna mess up our notes again?
Don't do it. If
I don't
you
even-
erase it, if you.
I don't even know what happened. I don't even know what happened.
I got a lot.
I'm gonna rip it.
Not even surprised, to be honest.
I don't even.
I think I'm just gonna start over.
I'm gonna take notes like 5 minutes after he writes it.
Oh. Does anybody have whiteout?
Gosh, that was terrible. I don't even know why I would write.
Because it doesn't make sense.
Yeah, it doesn't make any sense at all. I don't know why I wrote animals. That's so wrong.
We just took your word for it.
That's so terrible. I know you. Now this is back-to-back days. It had been so long since I made a mistake. It was like maybe the second week of class, the last time I wrote something on the board that was wrong, and then now it's back-to-back classes. Gosh, that is just.
You lured us into the safety.
You.
Trusted
I
you.
know you deserve better, and you haven't gotten better. I'm sorry. So glycogen. This is a glucose polymer.
I love how all the words have Y's in them.
Doctor,
What?
doesn't have a Y.
I like all the ones that like.
Yeah.
I'm sorry to quote Emma, but why are some people more like
What?
susceptible to having acne than others?
It has to do with the particular bacteria, like the particular strains of bacteria that live on the surface of your skin. We all have bacteria that live on the surface of our skin. Some strains of bacteria are more prone to cause inflammation than others. So what acne is, it's an inflammatory response, and so so that's why there are certain foods that will trigger it, because it provides more for those bacteria to eat and divide really quickly and then trigger that immune response. Yeah, it has a lot to do with the particular type of
So
skin
genetic.
bacteria that you have. It is. I mean, it's mostly genetic, but a lot of it is environmental as well. Because your skin, the particular type of skin bacteria that you have, is a combination of your genetic makeup and the environment that you're exposed to.
Sometimes I'll go surfing for a week and my skin clears up like so nicely.
Well, it dries out.
Oh, okay.
Yeah. So in the in that salty air, your skin dries out and the bacteria go dormant. A lot of them. Yeah, Lydia.
So this is random as well, but
I love it. Can I write as you? Is it
Yeah.
going to be rude if I write as you talk?
It's fine. So
I mean, it is going to be rude, but can I do it?
go ahead.
Yeah.
So
I would have said no.
I was
Oh, we know you.
Watching something and there was like this girl was born with something where she would like just as a lifelong she has to sleep like she falls asleep after a certain amount of time. And
Okay.
She can't control it. So like why is that? Like why yeah, she just keeps passing out.
I'm cooking meat.
Yeah.
She for like weeks.
Yes.
And her mom still wake her up to use the bathroom and eat. Yes.
Oh, shoot.
Okay. So why is that?
Her brain did not develop correctly.
That is my one.
Yeah.
Like what part?
I don't know. She probably has a very unusual mutation. That's a very rare genetic illness. A lot of genetic illnesses are extremely rare. So I don't know. I'd have to look at. I'd need to see the data. Emma and then Tori.
Why does stress cause acne?
Again, so stress usually leads to the production of certain, not neurotransmitters. What's the term I'm looking for? The release of certain hormones, stress hormones, and those stress hormones can trigger bacteria to grow more quickly. But most often, what it does is it triggers your immune system to behave erratically. And so it's why it's also a lot more common to get sick when you're stressed out and your immune system is behaving erratically. And so your immune system reacts more to things it shouldn't, like the presence of bacteria that have lived on your skin for ages. Tori.
Are you sure?
Yes, positive. Yeah, I got this all sorted out, and I will not make another mistake for for months. For months.
Is that a promise?
If I write something on the board that is wrong between now and the beginning of February,
You have to bring us donuts.
I will bring every single person in here individually what you want, up to a cost of $5.
No way.
Yeah. Yep. I'll do it. Okay. So glycogen, glucose polymer,
I
function,
love this. He's gonna do it by the end of class.
energy storage in animals. The other
Don't mess up.
carbohydrate polymer that you need to know is chitin, which is also a glucose polymer. Look at this, a lot of glucose polymers. And it makes you wonder, why is everything centered around glucose? What is it about glucose? It is C6H12O6, but so are many other sugars, are also C6H12O6.
I thought I was
So
smart.
chitin is a structural carbohydrate. So you find it in the carbohydrate.
Wait, what's it called? What?
Chitin,
Oh, I thought it was like
chitin, chitin,
Titan?
kitten.
Is it chitin or
I don't know. So chitin, it's a glucose polymer. By the way, my mistake has to be written on the board, by the way. Okay. So chitin, it's a glucose polymer that's structural. You find it in the cell walls of fungi, and you find it in the exoskeleton of arthropods, like insects, crustaceans, spiders, centipedes, millipedes, scorpions, horseshoe crabs.
She does.
Okay? All right. So these are carbohydrate polymers, and if they are a polymer, what are they made of?
many
Monomers. And in this case, the monomer for all of these that you need to know is glucose.
Wait, I'm sorry, can you repeat that? If they're what?
If there are polymers, they are a chain of monomers, and in this case, the monomer that they are made from is glucose. Okay? Do you know that there are 2 cells in your body that can only metabolize glucose? They can't metabolize anything else.
What are they?
So they require glucose in order to function, and they're the red blood cells and neurons. And you're like, well, how is it possible? I know what you're thinking. You're like, how is it possible then to do a low carb diet if your neurons that make up most of your brain can only metabolize glucose? It's a wonderful question. It's a wonderful question. One in which I'd like you to spend a little bit of time talking with those around you and try to come up with a solution.
Wait, I'm sorry.
So if your neurons, right, that carry out most of the functions of your brain, and your red blood cells, that kind of important, if they can only metabolize glucose, they cannot use any other energy source, how is it possible to function on a low-carb diet? How is it possible to function? Take a couple of minutes. I'm going to erase this. Did you all write this down? Because I'm going to erase it. And I'd like for you to take a little bit and try to figure that out, how that works. This is called critical thinking, by the way.
Okay, so I think because they had it like stored up already, maybe. And, like how do part like your brain and your blood only work off of carbs?
Yeah, so if your neurons and your red blood cells can only use glucose as an energy source, how is it possible to survive on a low-carb diet where you consume very little glucose?
You can't survive without carbs.
Or maybe they get like the other elements.
I mean, we're talking about surviving. We're not talking about living. I would agree that you cannot live without carbs, but you can certainly survive.
I'll say glucose.
Have you seen the crudes?
Yes.
Where Eep is like, no, this isn't living. It's just not dying. Right? So they live in a cave, but Eep, she doesn't want to live in a cave. She's the oldest child. Eep, I thought forever it was Eve. And then my daughter's like, no, Dad, it's Eep. And I said, no, Reagan, you're wrong. And then I went and looked it up. Sure enough, it was Eep. Oh, man. Anyways, she already knew she was right.
You
Reagan
can't even hate on her.
Reagan pretty much is almost always right.
She's so
7.
smart. She's so
But she's our oldest. She's smarter than I am though.
She's-
Yeah, maybe.
It was so funny, it was
All right. Do we have a solution?
Yeah.
Okay, what's the solution? Okay. Katie is going to give us a solution. Or is Carrie going to give it to us?
Katie.
Yeah, Katie.
Okay.
Yeah.
I'm stealing Carrie's idea though, but she says lipids because they can do the same thing as lipids can do the same thing as carbs pretty much.
For almost every cell, but
Yeah.
not for red blood cells and
Oh.
neurons.
Well-
They can't use lipids. They
Also,
can only use glucose.
isn't glucose and other things than just like bread? So
Uh,
for example,
yes.
like apples and things, you can still eat those.
You can, although you can still, if you're doing a low carb diet, you don't eat apples.
What?
You don't eat apples.
Yeah.
Why?
My dad can't.
Because if you're doing a low carb diet, you've got to do under 20 carbs. And an apple, I don't even know how many carbs are in it, but a lot. Oh, yeah. That natural sugar. Just because it's natural sugar and unprocessed doesn't mean.
Yeah.
What do they?
What about grapes?
Grapes. Still, you could, but still, it's like, where is this glucose coming from? Right? So if you're eating food that's that virtually has no carbs in it, you're not getting glucose in your diet, but that glucose has to come from somewhere because those cells can only use glucose for energy.
Photosynthesize.
Bingo.
If you're an animal.
Make your own cookies.
Okay, so here's the thing. There's a glucose polymer used for energy storage in animals. What is it?
A lipid polymer?
A glucose polymer.
Polysaccharide.
It's glycogen,
Oh.
right? Glycogen is a glucose polymer
Nerd.
used for energy storage. And so you build up a lot of glycogen in your liver that you use in between meals to get glucose into your blood so that red blood cells can eat, your brain can eat. But you tend to wear through that fairly quickly, like 12 to 20 hours, depending on how much glycogen you have stored up. Tori,
How are
glycogen.
children diabetic?
How are, because their pancreatic cells do not generate insulin the way they should. So they didn't get to that point by just, putting a lot of pressure on the pancreatic cells to make insulin for long periods of time by overindulging in sugary goodness. But instead they were born with something wrong with their pancreatic cells. So there are a number of issues that could trigger. It's called type 1 diabetes versus type 2.
And it's like, so type 1 is like you're born with it. Type 2 is
Yeah.
like you develop it over time.
Yeah. And so type 1 though could onset late in age. Like some people still don't start showing signs of type 1 diabetes until they're in their 30s, 40s, 50s,
Oh.
even though it's genetic.
So then how do you know the difference whether it's type 1 or type 2?
Because of the nature of what your pancreatic cells are presenting.
So
And then
that-
if you have pancreatic cancer and you have to have a good deal of your pancreas removed, it usually induces type 1 diabetes. It is.
So they
He's
say like when you eat a lot of sugar, that's how you get it, right? Or how's
Yeah.
that- That's it? Just,
Yeah, by overworking your pancreatic cells.
and does exercise help with that or no? Just-
Yes, I mean you can treat a lot of the symptoms of diabetes by being in better shape. Yeah. Okay. So the solution is there are chemical processes by which you can convert other molecules into glucose. So our bodies are capable of making glucose not through photosynthesis, but through other chemical mechanisms, usually by converting fats into glucose. It's true. It's called gluconeogenesis. Gluconeogenesis. All right. So lipid polymer. The lipids are weird in that most lipids are functional as monomers and they don't ever polymerize. Most lipids, again, are functional as monomers and never polymerize.
What's polymerize again?
Polymerize is to form a chain.
Oh, gotcha. Like the other one.
Yep. The only real lipid polymer are fatty acids. Or are fats. We'll just call these fats.
And we read about it.
And these have a glycerol backbone attached to 2 or more fatty acids. Okay? So lipid polymer, fats made up from a glycerol backbone attached to 2 or more fatty acids. You could argue that phospholipids are also a lipid polymer. They also have a glycerol backbone.
So the glycerol backbone makes it allowed to like form chain?
Yeah, that you attach the fatty acids and other material to that glycerol backbone, which is predominantly a hydrocarbon with some oxygen as well. Glycerol backbone with 2 fatty acids. True or false? Fatty acids are hydrophobic. True. With 2 fatty acids and 1 hydrophilic head.
What's hydrophilic is?
I like the water.
Yeah, water loving. So phospholipids have a hydrophilic head. And if you want, we can talk about the chemistry that makes it hydrophilic, or you can just say the hydrophilic head and leave it at that, since this is not a chemistry class. Okay. So these are really your only lipid polymers. The rest of our lipids are active as monomers. Like there's steroids, there are waxes, other hormones that are complex lipid monomers. Okay, but we'll talk about more of those in context. All right?
Yeah.
Okay, cool. Fatty acids come in 2 varieties. So we're like fatty acids, right? 2 or more fatty acids, 2 fatty acids, and a hydrophilic head. So let's talk about these fatty acids. They come in 2 varieties. 2 varieties. They come in the saturated variety.
I said phospholips.
What's that?
I said phospholips.
Phospholips.
That's unfortunate.
Phospholips. You left off the ID?
Yeah.
Sorry. Saturated. And what do you think the other form is?
Unsaturated.
Unsaturated. Look at that. Oh. Unsaturated.
Oh.
That was a
Hey,
spelling error.
spelling errors don't count. Hey, if you want to make spelling errors count, we can make spelling errors count. But then if it's going to count for me, it's got to count for you too.
It's okay, it's all good.
Okay, so saturated, and these, in saturated fatty acids, there are no double bonds.
Those are saturated have no double bonds?
Yeah, no double bonds. And we know what a double bond is, right? We have one of our type of isomers that we talked about, left and right, or cis and trans isomers that are like, where is the double bond, right? Where are the big bulky pieces with respect to the double bond? Okay, unsaturated then, these have at least one double bond. Okay? Yeah, Michelle.
How is the drawing look different?
You want to see how they look different? Or I was just going to do it. Yeah.
I know how to draw a phospholipid bilayer.
Do you?
Me too.
Everyone else.
Shut up, Bella.
Gosh, you guys are so mean to each other.
I was
It's mostly Emma.
I'm not mean.
See, I don't think it is mostly Emma.
I'm on Tori.
What?
Tori's more open than me.
I won't say I don't think it's mostly Tori. All right. So, saturated
Nick, you're so quiet in this class.
I know, what the
He's
heck?
not quiet in his
Oh,
other classes?
Oh my god, in Watson's class?
Really?
Really?
Math?
In other classes, he's the loudest one.
Intriguing.
I overpowered him. Like, a loud one.
That doesn't surprise me.
No, in
Surprise
other
me.
classes I'm pretty quiet, just this class.
This is true. Yeah,
That's because I don't feel comfortable
It's
with
true,
other
you are
people.
pretty quiet.
I don't
Okay.
feel
She doesn't
comfortable
say
with other
anything in
people.
Watson's class.
So remember,
I like
fatty acids. Fatty acids are just hydrocarbons. Okay. Fatty acids, they are hydrocarbons. So they are molecules that contain carbon and hydrogen. Okay.
It's pretty cool.
And so saturated looks like this. Remember, there are no double bonds in a saturated fatty acid. No double bonds. So what is it saturated with then? Because where do we use the term saturated?
Do you want to draw like for?
Oh, I do want, I mean, I really don't care whether or not you draw this. You will want to draw this.
So you
Again, I will stand by my original statement. I mean, maybe I should, but, you know. So somebody tell me where we use the term saturated. Yeah, Emma. Okay, liquids being saturated, say, with salt, right? Where you can't get any more salt to dissolve in it. Or the soil saturated with water, right? And then it starts sliding down the hillside. Yes?
Yes.
Okay.
That's a mess.
So what is this saturated with?
Hydrogen.
Hydrogen. It has as much hydrogen as it can possibly have, because check out this unsaturated. This one we're only going to do one double bond. Oh boy, indeed. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Good. I got the same number of carbons. I tried to make sure that happened, but wasn't paying super close attention to it. Okay. Same number of carbons on either side. Yes? 5 on the left, 5 on the right. How many hydrogen on the left?
12.
12. How many hydrogen on the right?
I think, well,
10.
10.
to us. Yeah.
Right? Would you agree that 10 is less than 12?
Yeah.
All right. We're in a good place.
We know math.
Okay? So 10 is less than 12.
Oh.
So 12 is as many hydrogen as you could possibly fit in that hydrocarbon. Yes? 12 is as many as we could fit. 10 is less than 12. This is unsaturated. It is not saturated with hydrogen. It's not holding as much hydrogen as it possibly can. Okay? This is cool. You love this, right? Okay. But we're like, why do we care? Why do we care? I don't know either. No, I do. Okay. What do we know about a double bond?
They share 2 electrons.
Yes, we do know that they're sharing actually 2 sets. So they're sharing 4 electrons. But what do we know? We talked about isomers. That there's some 3-dimensionality at this double bond.
Oh.
Yeah.
I'm so confused as to isomers. Is it like
Isomers when you have the same molecular formula
but different shape?
but different shape.
Gotcha.
Okay, so here at this double bond, we actually get a kink in the chain. So this would look like this. 1, 2, 3, 4, 5. Okay, this is another way in which we can draw this, and this is a more accurate representation of its 3-dimensional shape. I know this is only 2 dimensions, I got that. But this is a more accurate portrayal of its 3-dimensional shape. This one looks like this. 1, 2.
Oh, shoot.
1, 2, 3, 4, 5. Okay.
I
Are you
think
sure?
we qualify
Is it
as a
final?
mistake as like he does it, he tells it to us,
Yeah,
and then he comes back. And
Okay.
Oh wait.
So this right here, what type of double bond is this? Is this cis or trans?
Well, okay, what's the difference?
Cis, the bulky parts are on the same side of the double
What do you
bond.
mean the bulky part?
So this is the double bond.
They're,
So the bulky parts, because there's a hydrogen here that we didn't draw, and there's a hydrogen here that we didn't draw. But this bulky parts, this is a methyl group, right? One of our functional groups. This is a carbon with the 3 hydrogens. That's this. Is right here. And then this is these other 2 carbons with their hydrogens. Okay? So here's our double bond. Is this cis or trans?
trans.
Trans, right?
So what's trans and cis is when
The big bulky groups are on the same side.
so it's even.
Yeah, you can look at. I mean, in a trans, it's more even because you have a big bulky group on this side, and you're kind of balancing it out.
Okay.
But then, if this were cis, so let me erase these hydrogens because they're mucking up my drawing. Okay, so now let's do this. And now, what kind of a double bond is it? It's cis, right? Now it's a cis double bond. And look at this.
Glucose thing, right?
What's that?
Like it's making the glucose thing?
Yes, almost like a ring. But do you see that this is a kink in this structure? This is more linear. This is not linear. Okay, right? Linear, not linear. Which type of fatty acid do you think you can pack more tightly together? A linear or a not linear? Linear, right? It's like I could fit a whole bunch of pencils into a single box. Yes? Because they're nice and linear. Where'd that marker cap go? There it is. They're nice and linear. You can't fit as many of something that's like a weird odd shape in a box as you could pencils, right? Unless of course their shapes were just nice and like almost like a jigsaw puzzle where they fit together nicely. Okay, so these we can pack in really tight. These we can't pack in as tightly. Okay? These tend to be solid at room temperature. These tend to be liquid at room temperature. Solid,
Saturated?
liquid, yes. So saturated fats tend to be solid at room temperature. What are some fats that are solid at room temperature? Butter, right? Oh, I love butter. It's so good. So good. And what's a fat that is liquid at room temperature? Emma? Oil, like vegetable oils, coconut oil. And so animal fats tend to be saturated fats. Okay? Animal fats tend to be saturated fats and solid at room temperature. Right? So you think of like the fat on bacon, right? It's solid at room temperature, right? You heat that up. Now it becomes liquid, gets you that nice little bacon grease, but you put that bacon grease in the refrigerator,
Ew, no.
and it's. You don't keep bacon grease?
No.
I keep the bacon grease. Because like if you're going to make some scrambled eggs one day, but you're not going to cook bacon, do you want to make them in butter if you can make them in bacon grease?
We keep
No.
it, but then we throw it.
Sounds unhealthy.
I put it in my dog. Keep it so it like solid.
No, we keep
Oh, and then you throw it away? Oh, I keep it so I can like scramble eggs with it. Or, so do you know what mayonnaise is made out of? Egg yolks and some fat. Some additional fat. Egg yolks aren't fat, but some additional fat. So you can make mayonnaise with egg yolks and bacon grease.
Okay.
And then it's baconaise. Yeah, Isa.
There was a girl in my elementary school who would take the little packets of mayo and just straight up eat them.
That's
So
gross. Yeah, that's gross.
Psycho.
Okay.
If I'm eating a sandwich and it like comes out.
So this is it.
Really? Actually, cool.
Do soy sauce chicken competition at our school.
All right. Let me.
I feel like that's something I would win.
All right. We're recording again. So you're the mutiny you're staging. There's documented evidence of it now. Okay. Look up here. Look up here, everyone. Does the order of letters matter? Really?
Yes, it does.
The
So
alphabet,
Yes, it does.
technically, the order of the alphabet really does not matter.
Well, don't they share the same structure?
For what?
No, it doesn't. Well, yes, it does.
Why does A have to come before B? Why?
I'm not talking about the alphabet in general. I'm talking about, are these two things the same thing?
No, because one's A and one's B. Stop doing things.
Shh. They're made from the same pieces, but they are not the same thing. You and I are made from the same pieces, we are not the same thing.
We are.
Emma.
So are you saying like when you get in the front there, you're taking it from the last like chain of all things?
I'm just saying that these two polypeptides, these two amino acid polymers are different. This one A comes first and then B, this one B comes first and then A. Yeah.
Okay, but don't A and B have the same structure?
Well, they would have different R's, which means they potentially have very different
If the
chemistry.
R's are different then.
Correct. Yeah, if they're the same. Yeah, if these two things were the same amino acid, then it would be exactly the same. But that's why I did different letters. So in this case, we have 10 different amino acids. Now, what would have been really cool is all 20 amino acids have a single letter of the alphabet that you can use to represent those amino acids.
Even Z.
No, well, there are 26 letters and only 20 amino acids.
Oh.
So some of the letters aren't used.
Let's use Q.
So if A and B were the same amino acid, then these would be identical, yes? But if they are different amino acids, these are not the same thing. Would you agree? A, B, C, D, E, F, G, H, I, J is different.
So it doesn't have to be the matter.
The order matters. These two amino acid polymers are different. This right here, this is the primary structure. The primary structure is the order of acids. Okay, that's the primary structure of a protein, is the order of amino acids. Okay? And again, these two are different, right? How do you know they're different? Because they look different, right? And then what you would have is they would behave different chemically as well. But you just look at them and they look different. If you can tell them apart, they're different, right? True or false? You can tell me apart from anyone else in this room. That's, you think Kyle and I look so similar? You cannot tell us apart. Kyle, I am so sorry that they would say
Hey,
that
will you
about
ever
you.
shave the beard?
In my entire life, probably.
Like
I haven't set a date on my phone.
what would you
Or-
look like if you shaved your beard?
Oh, I look completely different.
Oh, I can't imagine. I bet you would.
How do
Okay.
you groom it? Do you use scissors and just like trim it?
No,
Do you have beard oils?
I do, but I rarely use them. I only use them when I go to church. So when I go to church, I oil my beard and comb it up a little bit. Yeah, you gotta church it up a little bit. What's that? Okay. So, well, now I'm all distracted. So this is the primary structure. Right now, we haven't talked anything about the three-dimensional shape of this protein, right?
Correct.
Now we're going to start getting into our secondary and tertiary structures.
This is for Kyle.
Yeah.
Got it.
Okay. So we know what our primary structure is, yes? It's the order of acids. Okay? The order of amino acids. Our secondary structure are regional shapes in the polymer. Meaning they're not spanning the entire polymer. They're just certain regions of the polymer have a particular shape. Regional shapes in the polymer. Okay, regional shapes in the polymer. Okay? That's the secondary structure, regional shapes in the polymer. There are two very common regional shapes. Something that looks like this,
Curly fry.
and something that looks like this.
Regular fry.
Okay? This is an alpha helix, and this right here is a beta sheet.
I love
an alpha helix, and a beta sheet. Which of these do you think is more common based on the order of the Greek letters?
Alpha.
Alpha, right? I don't know if you knew this or not, but the Greek alphabet is alpha, beta, gamma, delta, epsilon, and then I don't know what happens after that.
Lambda.
Yoda?
Yoda.
Iota. It's the Greek equivalent of I, not Yoda.
Iota.
Iota. The Greek equivalent of I.
One of my favorites is delta.
It's pronounced the same way as an I. So,
Sigma? No.
Sigma's no good.
Lambda's my favorite.
Lambda's a good one. All right. Okay. So now we have primary and we have secondary. If secondary is regional shapes, what do you think tertiary is?
Not
Not
regional.
regional.
So tertiary, and I know this is just kind of all over the board. I don't know. Do you try to copy the shape of the notes as well, or do you just write them in a sequence?
I kind of depend. I write them in
I write them.
Okay, so tertiary is the global shape of the polymer. And in the same way that the globe is the entire Earth, the globe is what? It's the entire protein. Oh my goodness. Oh my goodness.
Wait, what is it? Global shape of polymer?
Of the polymer. Secondary structure is the regional shape in the polymer. The most common type are alpha helices and beta sheets.
That's for secondary?
That's for secondary. Tertiary, there's no, I mean, you can't even say what's the most common shape because every protein has a different shape.
Yeah.
Every one. And it's the global shape of the polymer. All right? Questions? Thoughts? Concerns? Why?
Is it break time?
It is break time. So I'm gonna pause this. Yeah. And just okay. Absolutely. So up on the board we have probably the two most well-known examples of eukaryotic cells, not among scientists, but just among biology classes. You tend to learn two types of eukaryotic cells. You learn animal cells and you learn plant cells. Okay? Now, these are both examples of eukaryotic cells. What does that root mean?
many.
No.
Is it a Greek? It must be a Greek root if you don't know it. Because if it was Latin, then you ought to be able to fire that off. Oh, you would.
I would know.
Okay, so it means true.
What means true?
U. This root EU, which is intriguing because my middle name starts with EU. True gene. All right. Anyways, well, it's interesting. Eugenics is an effort to get rid of all bad genes. It's basically it's getting rid of, not allowing people to reproduce because you want the true genome. And I think that's where the origins of Eugene comes from, which is a little disturbing. Anyways, so eukaryotic, these are cells that have a true, and this karyos is nucleus. So these are nucleated cells. These are cells in which the genetic material is packed away in the nucleus. So inside of these nuclei, we have DNA.
That looked really good.
Thank you. That one is not as good.
That looks better.
I got so excited that you complimented my DNA. I actually tried on the second one and didn't produce something as good. Inside of the nucleus. Inside of the nucleus. Yeah, Michelle.
So in both the animal cell and the plant cell, the nucleus is the big cell.
Yes, although in a plant cell, you find this was a structure left off. You find a massive central vacuole in the middle of a plant cell that functions to maintain pressure against the wall of the cell wall. And so you've got a massive central vacuole full of water. That tends to be the biggest structure in a plant cell. But yeah, you can still find the nucleus fairly easily.
Well, what is the small
Which one? Oh, inside of there? That's called the nucleolus. Yeah, both of them actually drew it. It's called the nucleolus, and it's where you are transcribing ribosomal RNA. But we'll get there. Anyways, we'll get there following Thanksgiving break. Again, Thanksgiving break. It's going to be Dr. Engel's story time.
Yay.
It's going to be Dr. Engel's story time. We're going to walk through the story of a cell. A little bit different than the textbook does, but it's fun. It's fun. It's fun. Now, I have to tell you, I didn't write it,
You should write a book.
but it's a fun story. Okay, so we've got the nucleus. These are eukaryotic cells. Prokaryotic cells do not have the nucleus. Pro is the root. It means first or the beginning, and the beginning nucleus doesn't really make sense. It does not have a nucleus. Okay, prokaryotic cells lack a nucleus, and what they look like is basically this.
There's random stuff.
Okay, where the DNA is, first of all, it's not linear. It's not forming linear chromosomes like we find here. You see this nice line of DNA. It's not packaged like that in prokaryotic cells. By the way, these are not to scale. Prokaryotic cells are tiny compared to eukaryotic cells. And it's not packaged in linear chromosome. It's just, it almost looks like spaghetti. Just piled in there, but without the sauce. Like just the noodles. And then, so you've got DNA. You still have ribosomes.
Wait, what are we drawing right here?
This is a prokaryotic cell.
Oh, okay.
These are both eukaryotic cells. This is plant. This is animal. Okay? In a prokaryotic cell, you still have DNA, you still have ribosomes. And you still have a plasma membrane. And so something that you find in all living cells, whether it is prokaryotic or eukaryotic, you find 4 features that are present in all of those cells. The membrane. Where's the membrane, right? We see that in all 3 of our cells. DNA. It's packaged differently, but there's DNA in all living cells. Ribosomes, yes? And then there's a 4th one. And what is it? Yeah, Rick.
Mitochondria.
Nope, you don't find mitochondria in prokaryotic cells. Remember we talked about with the whole eukaryotic from prokaryotic, the story that we tell is how you got the first mitochondrion,
Yeah.
right? But it really doesn't tell us where you get the first eukaryotic cell. It just tells us where you get the first mitochondrion. Emma.
Is that like a liquid?
Yes!
Oh, what's that called?
Yeah, the liquid part of the cell. It's called the cytosol. Cytoplasm.
That's it.
That's
what I.
Oh, so I don't even know. I'll just write it over here. Cytosol or cytoplasm. So the cytosol is the fluid. This is the fluid of the cell.
I was thinking,
And you're
about it,
like-
I just didn't know the name of it.
You're like, Dr. Engel, isn't it just water? And it is, but with all of the materials of the cell dissolved in it. And it actually is semi-solid. It's semi-fluid, semi-solid. It's like a gel. So the cytosol is the fluid. We call this structure the cytoplasm. Is the structure. So the actual fluid is called cytosol. The structure of it is the cytoplasm.
Structure of what?
The fluid. Yeah. And so, listen, I don't want you to think of a cell as just like a lake, right? Where you've got water, and if you want to move on a lake, right, you just row your little boat. Like, why would we row the boat, Dr. This is 2018. I'm gonna put a massive engine on my boat and go wherever I want to go. Okay? But that's not the way. That's not the way you should think of the cytosol. You should think of the cytosol as an incredibly complicated underwater city
Like Atlantis.
Yes, like Atlantis. Or where does Ariel live? What do you call that underwater kingdom? I know she lives under the sea, but is it Atlantis?
I've never seen that movie.
Your child is silly.
Is it?
I think it's just King
Yeah.
Triton's kingdom.
King Triton's kingdom. All right. So that's the idea I want you to have of this fluid component of the cell. Okay. Not that it's like a lake with fish and other things in it, but it's a really complicated, highly structural underwater city that materials are in and being transported in very directed and focused ways. And then I'll share you again. We'll come back from Thanksgiving break. It's going to be a long time before we see each other again, except for the 7 of you that I have parent-teacher conferences next week.
You don't have.
But
You have 4 of us.
other than that,
4.
It's me.
4.
I
No, it's
care
7.
about.
No, it's 4.
No, it's
You
4.
only named like 4 last class.
No.
Yeah,
I
you
named.
named much more than 4.
No, I named all 7.
You said.
You said
Well, y'all are true.
So it's
You said 4.
There were 7.
Emma, Rick, Tori,
You didn't
Issa.
name me last time.
I did. And then Michelle, Carrie, and Mark. Okay, anyways. So other than the 7 of you that we have parent-teacher conferences, this is the last class we're going to have until the Monday following Thanksgiving break. And when we come back, we're going to do this activity, but through a story. And it's going to be incredible.
You should write a book like not just adopted.
All right? What do you think
About
It's
what?
coming back. This
about
is
all
about-
that?
Oh yeah, I remember this kind of stuff. I remember doing it.
Chapter 2 through 6, I don't know.
There's one more thing that I want to go over today before we finish.
And what is that?
And what is that?
Yeah.
Okay, so we already talked about the 4 components that you find in all cells, whether they are prokaryotic or eukaryotic, right? And so the last thing I want to talk about is focusing in on eukaryotic cells. I want to talk about what benefit does having all of these organelles give the cell? Okay? And so as we look at this and we look at a eukaryotic cell, you notice right away it's got a nucleus, right, which is where the name is from. True nucleus, first nucleus, right? We've got a nucleus, the DNA is packaged in there in a nice, beautiful linear structure or a bunch of linear structures. Here it's just kind of packed in there like spaghetti. But you notice there are a lot more structures that eukaryotic cells have in addition to the nucleus. Yes? That you don't find in prokaryotic cells. In fact, you only find, not counting the DNA and the cytosol, you only find 2 structures in prokaryotic cells that you find in eukaryotic cells. Ribosomes and the cell membrane. And so I want you to think about this for about 30 seconds. And I want you to tell me 3 benefits of being structured like this. Issa.
Where do you find the prokaryotic cell?
Where do you find them? These are bacteria and archaeans, but bacteria.
What about a human cell?
So there are no prokaryotic human cells. Human cells are all eukaryotic,
Yeah, but what are those?
like animal cells.
Okay.
But then we have like neurons nerve cells look completely different than skin cells.
Please excuse interruption. This is for all student drivers. If you are a student driver, please pay close attention. If you drive a gray Chevy Bolt, we need you to meet Mr. Culp at the upper school office area, a gray Chevy Bolt, immediately at the upper school office area. Thank you.
I said, is it a good thing or a bad thing?
Probably bad thing.
Who drives it?
I drive a gray Chevy Bolt.
Oh, shit.
I totally do. Yeah, but anyways. All right. I am dead serious.
Definitely.
Yeah.
You.
Now I'm trying to think of.
Where's your park?
I parked over by Greg.
That's probably.
That's probably you.
Because that's the student section.
Is it?
Yeah.
It doesn't matter. I drive a big fat car. I park in the teacher section.
Okay. Anyways,
I don't know.
do you guys have like carts, parking permits? I wonder if somebody hit my car. That's fine. I don't care. All right. So prokaryotic. All right. So while I go and meet them at this upper desk area, come up with some benefits of this. What time is it?
You gotta let
It's
us know what it
9:38.
is because I'm kind of scared for you.
Oh, what happened? I thought you wanted the answer to